Hello everyone, I am Ritesh and today the topic of my presentation is subretinal implants. First of all, I would like to go through the overview of my presentation. Firstly, I will discuss about the motivation behind the subretinal implants. Then I will introduce you to some basic anatomy of the eye and the cellular anatomy of retina and different type of cells that make up the retina. After going through this, in the second section, we'll go through the different type of retinal implants and understand why we are interested in subretinal implants particularly. In the last section, we'll discuss some state-of-the-art developments in subretinal implant technology where we'll see the Alpha IMS by Retina Implant Germany, the photovoltaic retinal prosthesis which is being developed at Stanford University, and some novel arrangement of electrodes and the techniques in order to see a continuous image just like a human eye would see. So first, let's discuss the motivation. Retinitis pigmentosa and age-related macular degeneration are the diseases that affect photoreceptors of retina and cause progressive vision loss in around 4 million people worldwide. AMD is the leading cause of blindness in people with 65 years of age. The degeneration of macula results in loss of central vision. On the other hand, retinitis pigmentosa is a progressive eye disease. It damages the rods and cone of the retina and it is caused genetically. The rods are attacked first, which cause the loss of peripheral vision, as we can see in the image. As the stage progresses, it affects the cones and the person may end up in complete blindness. The visual prosthesis, which we are going to discuss in this presentation, aims at partial restoration of visual functioning using electrical stimulation of remaining retinal network. It has been found that even after the degeneration of rods and cone cells, there can be inner retinal nerve cells that continue to function several years after the loss of rods or cones. So let's begin. Firstly, let's see about how the human eye functions and let's see the anatomy of human eye. So first of all, there are two kinds of photoreceptor cells which are rods and cones. These are present inside the retina. The rods are responsible for seeing the visualizing in dim light condition, whereas cones are responsible for the perception of color and sharp images. In the front of the eye is cornea, which is the transparent structure that covers the iris and the pupil, and it is the first primary focusing structure of the eye. Behind the cornea there is lens, which is another transparent structure. It helps to focus the light and form the image on the retina. Macula is the central portion of the retina which is responsible for formation of sharp and clear straight ahead vision. Sclera is the tough outer covering which protects the eyeball, and iris is the colored ring tissue which is present behind cornea that regulates the amount of light entering the human eye. Pupil is the small opening in the center of iris through which light enters the human eye. Optic nerve is the bundle of nerve fibers at the back of the eye that carries the signal from retina to the brain. And lastly is retina, which is the light sensitive layer of tissues that lines the back of the eye. So let's delve deeper into the structure and anatomy of retina. Retina primarily consists of three types of cells: the photoreceptor cells, the neural cells, and the supporting cells. So the photoreceptor cells are of two types, as we have already seen, the rods and the cones. The rods function in the dim light condition and are responsible for black and white vision. Typically, a human eye consists 120 million rods, and they are located in the peripheral area of retina. The cones work best in well illuminated conditions. A typical human eye consists 6 million cone cells, and central retina is dominated by cones. After cones, there are four different types of neural cells. Firstly, the bipolar cells. These connect the photoreceptors to the ganglion cells, as we can see in this image. The ganglion cells are the cells that finally become nerve fiber and end up in optic nerve and end up in the brain and carry the signal from eye to the brain. Then there are amacrine cells which connect the bipolar and the ganglion cells with each other. These are located horizontally, as we can see in the image. There are 40 different types of amacrine cell with each having its own neurotransmitter and has a particular function. And lastly, there are these horizontal cells which are responsible for connecting the bipolar cells to each other and are responsible for our eye to distinguish from dim and bright light conditions. Lastly, there are supporting cells which are of three types: the Mueller cells, astrocytes, and microglial cells. The Mueller cells maintain stability of retinal extracellular environment by regulation of potassium ion concentration. The astrocytes act as guide and support systems. They also are highly permeable to potassium and absorb excessive potassium and clear it from the surrounding environment. Lastly, the microglial cells are responsible for maintenance. They constantly scavenge for plaques or damaged neurons and synapses and infectious agents. As most of the antibodies cannot cross the blood-brain barrier, the microglial cell absorbs the unwanted plaques or damaged neurons and pass it on to the blood so that antibodies can take care of them. So after studying the structure of retina in the human eye, we'll go to the type of retinal implants which can help in diseases like retinitis pigmentosa or AMD. So firstly, we see epiretinal implants. We can see in the image that the photoreceptor cells have been damaged. This in the front is the epiretinal implant. The device is implanted onto the innermost layer of retina that contains the ganglion cells. So this device receives electrical impulses which contains the information of the image. So there is a camera which converts the image into some electrical impulses, and those electrical impulses are directly transferred using radio frequency to this epiretinal implant, and this generates electrical impulses which the ganglion transmits to the brain. The second type of retinal implants is the subretinal implants. In the subretinal implants, the device is implanted between the pigment epithelial layer, which is here, and the outermost layer of the retina, which contains the photoreceptor cells. Thousands of light-sensitive microphotodiodes equipped with microelectrodes are assembled on a very thin plate in subretinal space. So the light falling on the retina generates a current in the photodiodes and then activates the electrodes, resulting in activation of sensory neurons. So now, let's see what are some of the advantages of using subretinal implants. So as we saw that the microphotodiodes directly replace the photoreceptors, while the retina's remaining neural network remains intact. So all the signal processing is still being done by the neural cells of the eye. The surgery which involves the positioning and fixing of microphotodiodes is relatively easier, and there is generally no external image processing which is required as in case of epiretinal implants. And the other main advantage is that the eye movements can still be used to locate objects, which gives the patient a more better, which is similar to a human eye vision. Now, let us see some of the latest developments in subretinal implants. Firstly, we'll go through the Alpha IMS, which has been developed by Retina Implant Germany. In the figure we can see the implant. The tip of the implant contains 1500 light sensitive elements, and then there is a direct stimulation field for electrical simulation. In the magnified figure, we can see 4 magnified elements, which is each contains a 15 cross 30 micrometer photodiode, that that controls a diff amp, which is a differential amplifier, and a titanium nitride electrode. So when light falls on this chip, an image is captured several times simultaneously by all the photodiodes, and which generates a monophasic anodic voltage pulse at its electrode. Thus, each element forms a pixelized repetitive stimulation is delivered to the adjacent bipolar cells, which amounts in current dependent brightness at each photodiode. Apart from the microphotodiode array, there is a 4x4 direct stimulation test field as well. So this test field is required for interface characterization during the initial stage when the patient is just the implant. This direct stimulation field is used to determine the voltage threshold for perception and accustom the patients to the electrically invoked visual impressions and test the retinal excitability and spatial resolution as well. So in the first part we can see the implant. This is the fixation pad, the reference electrode, and this wire which comes out from the this comes out and gets connected to a plug which is the power supply. In this figure we can see this more clearly. So when from an object light falls on the object which goes directly to the human eye, the microphotodiodes, they generate electrical impulses which are carried to the bipolar cells and ganglion cells, which process the image and then transfer this to the optic nerve. Since the CMOS chip requires power for the diff amps and to take the image, the power has to be provided externally. This is Micah, one of the several successful patients who have been implanted with this chip. So let's see one of the tests that sorry
something round or no? I only see two objects. Only two. Okay, and so it's round on the left, and then the one on the right looks like it's longer and it's curving like a little bit like this. It must be a banana. I mean, that's what it looks like. Round. No. Apple. No, that's the very center of the shape that I can see.
So we saw how Micah was able to identify between the two different objects after being implanted with the chip from Retinal Implant Germany. So in the previous method we saw that there was a need for external power supply. So at Stanford University, Professor Daniel Palanker and his group have proposed another subretinal implant technology, which is a photovoltaic retinal prosthesis. In this, the photodiodes are directly powered through near infrared light pulses instead of getting supplied from a battery bank. So whenever we want to see an image, the image is first captured by a video camera located on the video goggles. This image is transferred to a small pocket computer which processes the image and converts it into pulsed near infrared light. Now this light is just projected on the LCD TV which is present in this video goggles LCD screen, and this image then enters the human eye and then falls on the photodiodes which are placed subretinally. So the near infrared light provides sufficient energy for these diodes while remaining invisible to any remaining photoreceptors. So there is no wire which has to come out from the sclera of the eye and connect to the battery bank. All the power is being supplied by the near infrared light. We can see a video in which we see how this operation is done.
Because the photovoltaic implant is thin and wireless, the surgical procedure is quite simple. As in conventional subretinal surgery, the procedure involves local retinal detachment by subretinal injection of fluid. A small retinal incision is made in the blade, through which the implants are inserted into the subretinal space. Several arrays of about a millimeter in width can be inserted through the same retinotomy to tile a large area. The retina is reattached after the implantation. The patient wears video goggles containing a camera, captures images of the visual scene. The pixel receives power media and pulls the radiance projected onto the retina. Infrared light, ultraviolet, converted by each photovoltaic joint pulses of light between the infrared joint. Succeeding neural signals propagate through the retinal network to the tissue, thereby restoring.
So after seeing the two different types of subretinal implants, let us also see a novel method for arrangement of electrodes. As we saw that the microphotodiodes stimulate the neural cells using some electrodes. In order to deliver the stimuli to many electrodes simultaneously, this ASIC or a application specific IC has been designed. In simultaneous simulation, multiple stimulating electrodes and return electrodes are used at one time, opposed to one stimulating and return electrodes. We can see in this figure there are hexagonal electrodes. The one in the black is the stimulating electrode, while on the side are the guarding electrodes. These electrodes are connected to sink and sources. Each electrode can be connected to a sink as well as to a source, which can be switched using an ASIC. We can see in this figure, in the first figure, we can see the center electrode as the stimulating electrode, while the surrounding six are the return electrodes. In the same arrangement, if we want to form an image in the retina at a slightly different spatial location, the ASIC can move the stimulating electrode from this to the one at the right, which is shown as A, which is shown here, and this now acts as a source and the other electrodes will act as a sink. This type of arrangement of electrodes helps in forming continuous image, which is very similar to what a human eye, the continuous image that a normal human eye would form. Thank you.